Good afternoon. It's good to be with you this afternoon. Thank GARM for the opportunity to come and share some of the work that we're doing in Paris and in Philadelphia. My name is Dan Chung. I'm the CMO of SparingVision, and today we're just going to share some of the work that we're doing there. So for us, SparingVision is a genomics medicine company totally dedicated to the eye. And retinal diseases really affect millions of patients worldwide. We've all heard of dry AMD and AMD, wet AMD, and this comprises almost 20 million patients in the US alone. When it comes to that, about 90% of those patients are in the dry form, and some of those will have something called geographic atrophy, which encompasses about 1 million. And then, of course, something that's dear to my heart, and my roots are in inherited retinal disease, and the whole Luxturna development is inherited retinal diseases. These are relatively rare, but altogether they make about 200,000 patients, both in the United States and in the EU. Of course, there are the vascular diseases, diabetic retinopathy, and the other vascular diseases that also have a sizable number. And for us, the retina is really the target area, although we're not opposed to the anterior segment as well, but we're concentrating in the retina. Somebody did tell me there was another part of the eye other than the retina, but that's where we're concentrating right now. SparingVision has 3 basic pillars. One is a gene therapy pillar, where we're basically looking at gene-agnostic ways to treat retinitis pigmentosa. We're also looking at the possibility of making this modality available for dry AMD. And then another program we're looking at that's actually restorative. The first one is SPVN-06, and this is hoping to slow down the progression of disease, whereas SPVN-20 is actually about restoring dormant cone cells, in other words, bringing back function in cells that don't have function anymore. Eventually, the idea is combining some of these transgenes together so we have a restorative effect, then a protective effect on top of that. Our second arm is something we're very excited about as well, and this is our collaboration with CRISPR Therapeutics. We have 3 targets that we'll be doing gene editing for in the ocular space, and those are moving forward. And the other thing is we're always looking at how to advance the field, and whether it's technologies or indications or things of that nature. We're very opportunistic. So there's a traditional look at our pipeline. The first one, SPVN-06. This is actually in phase 1-2 studies. We're currently in our dose escalation. We entered into our 3rd and final dose recently, and that is ongoing. SPVN-20 is the restorative one, and we hope that this will be IND preparedness and enabling, or basically applied for by the end of the year, and we hope to be in the clinic early part of next year, as well as the others that are in preclinical and the 3 products that we are doing with Intellia Therapeutics. So our CEO, Stéphane Boisel, has really assembled a group of veterans in the industry space, those that have gene therapy backgrounds or ocular backgrounds, in some cases both, 2 of whom are with me today here. Our founders are José Alain Sahel, who was at the Institute of Vision in Paris, now is the chairman of ophthalmology at the University of Pittsburgh Medical Center, and his collaborator, Thierry Livillard, who was at the Institute of Vision. Unfortunately, he did pass away this past year, and then Denise Dalcara, who was also at the Vision Institute. So for our board is led by Joe Papa, who was the former CEO of Bausch & Lomb, and we have a number of folks from the venture capital world, and then some that are also part of venture capital but have somewhat of a unique niche. We have somebody from the University of Pittsburgh Medical Center Enterprises, an academic institution with a venture fund, as well as the RD fund from a patient organization known as Foundation Fighting Blindness, and they are instrumental in moving us forward. We've raised about $135 million in Series A and B, and but we're always open to hearing about more opportunities if you'd like to chat. So when we think about retinitis pigmentosa, this is basically a schematic of the natural history of disease. And the reality is that retinitis pigmentosa, there are over 300 genes in the IRD space, and retinitis pigmentosa makes about 70 to 80 genes. So the reality is, and I bleed RP65 Luxturna treatment. I spent 16 years on that product. But we can't develop 300 different gene therapies for all these rare diseases. So we look at gene-independent ways of trying to treat some of these diseases. And so for RP, you have two specific cells, light-sensing cells. I'm not going to give you a whole ophthalmology lesson right now, but just keep in mind the photoreceptors are the light-sensing cells. There's something called rods that are on the periphery for dim light, and then central vision is the cone cells. That's for your best visual acuity, color vision, daytime vision. Right now you're using your cones to look at that screen or to look at me. And the idea is, can we just preserve this cone function in these RP patients for another 20 or 30 years and basically give them functional vision for their whole life instead of going blind at age 50 or 60 or older? So the idea is, can we treat these patients in a gene-agnostic manner? And as you can see, it's a progression. The rods die first, then the cones die, and then the cones become inactive, and then they actually disappear as well. So we're kind of sandwiched in between the gene-specific programs and then the optogenetics and the stem cells, which are a little more late in the progression of disease. So we're situated right there in the middle for the 06 program and for 20 as well. Now, the 06 program is based on the fundamentals of what we call rod-derived cone viability factor. And so the question really came up is, in these diseases, we know why the rods die because all the genetics involves the function of these rod cells. But then why do the cones die, the central cells? Because there really is no genetic correlation there. And this is where our founders really came up with this and identified this factor called rod-derived cone viability factor. And this is part of the NXNL1 gene. And basically what happens is that two different isoforms. One is secreted by those rod photoreceptors. It binds to the cone photoreceptors and starts a physiological process that maintains the cone function and structure. And then the other isoform acts as a powerful antioxidant. So that is a mechanism that happens in my eye and your eye as we speak. It's an innate process. We're not reinventing the wheel here. But the gene-independent part comes in is that it doesn't matter why the rod cell is no longer there. Whatever genetic mutation is or whatever is functioning, the cell is not there to produce that RDCVF factor. And with gene therapy, we can put that back in and try and jumpstart that protective effect in these RP patients. So the idea here is that we would take SPVN06, both different transgenes, we'll put them into one AAV, and we'll put them into a subretinal injection. And this is the way that Luxturna has done it, and most gene therapies have done it. Right now, it is a standard of care in many ways for gene therapy to deliver it. I know there's a lot of work in IVT and in suprachoroidal, and we'll talk about IVT because there are definitely places where intravitreal injection is probably better suited. So this is some of the preclinical data where you see here is in the blue bars, these are the animals that were treated with the RDCVF, whereas the others were treated with vehicle. And you see that there's a significant slowing of the degeneration of visual function. In this case, we're looking at cone function in these mice. This is done with an optokinetic response. And then the photographs of the flat mouse, you see a higher density of cone cells, which are depicted in red, compared to those that were treated and not treated. So this really gave us evidence that there is a slowing down effect when it came to RDCVF. This is the clinical trial. And the clinical trial, I asked my team to go ahead and name clinical trial because I think that's the kind of thing to do these days. And they came up with Prodigy. They told me it was a rock group. Well, I got to tell you, I've never heard of Prodigy as a rock group, so it tells you how old I am. Anyhow, so this is Prodigy. It's divided into two parts. There's a step one part, which is a traditional dose escalation, 3 doses, 3 patients each. In between, there's a DSMB meeting who gives us a full review and tells us to go on to the next dose. After that is completed, we'll have another DSMB meeting in which they will discuss the 3 doses and determine 2 of the best doses to go forward. This will then go to an extension phase, which will be randomized with a naive control group. At this point, we'll hopefully be able to enroll all these patients by the end of the year, and then there'll be a 1-year follow-up. Well, obviously, primarily, we'll look at safety, but obviously, we're looking at a lot of efficacy endpoints. You name it, we're probably doing it, to look to see what kind of efficacy we might be able to see after 1 year. And we have 2 sites right now, one at the University of Pittsburgh Medical Center in Pittsburgh and the Cansvan Hospital in Paris, and we'll be expanding in the United States to 4 other sites as well to enhance enrollment. I have to say that, I'm used to giving medical talks and clinical talks because I'm a CMO, not the CBO or the COO. I did forget to mention one thing. I will plug our company is that we were named one of the Fierce 15 last year when it came to biotech companies that we're extremely happy with on that. So one of the things about the patients that were treated, and the 6 patients that we did see, we see that there was extremely low inflammation scores in the ocular components. And these are all at level of +1. We don't get excited as ophthalmologists. I don't get excited until I see a +3 or +4. And these are all self-limited, so we had a very good score when it came to inflammation. The clinical trial had minimal adverse events, all related with the surgical procedure. Now, this is a little maybe disconcerting when you first look at it, but this is basically systemic immune response on the total antibodies against the capsid in the serum. And the smaller line is from dose cohort 1, which was the low dose. We see a small rise, comes right back down to where it should be in minimal detection. We only had time to collect data for the first patient of cohort 2. We did see a sizable gain in the response initially, which we expected because we did a dose that was significantly higher. But we anticipate this also will come down quite nicely in the future. And there was no T-cell mediated response to either the capsid or the transgene itself. Moving on to SparingVision, now named Nirvana. I did hear of that rock group. This is where we're actually trying to reinstate a phototransduction cycle. Phototransduction is simply those photoreceptors taking the light coming into the eye, translate it into an electrical response, and send it to the brain. As much as I would like to say the eye sees, the eye doesn't see a thing. It's the brain that sees. And without that signal to the brain, there is no vision. So we're trying to get these dormant cells that aren't functional anymore, try and put something called GRK, G protein, inwardly rectifying potassium channel gene, reinvigorate that phototransduction and get patients to have light stimulation or better. And this is basically a schematic of that. So here on the top is a healthy cone with what we call these outer segments. There are these discs that are constantly shed. This is a very dynamic area. Well, in dormant cones, we don't see that. There are no outer segments. And so the idea is we can use the machinery that is left with the GRK gene and then reestablish a phototransduction cycle, and hopefully patients will get light sensitivity or potentially acuity and color vision back. So with that, this is a little different because this is going to be an intravitreal injection, and we'll show you why, carrying the transgene of interest. And that is, when you get to end stage, and we'll come to that in a slide, the cells that are remaining in a certain location. Now, this is some of our preclinical data that we actually saw. ERG, if you don't know what ERG, it's like an EKG for the eye. So you know that a flat line in an EKG is not good for the heart. Same thing for the eye. A flat line is not good. So here we were actually able to restore the ERG signal, which is the electrical signal of the retina. And the OKT is just a visual stimulus test that we were able to regain. And also you can see that there are high levels of protein expression in the cones after the transduction with the GRK vector. So here is the whole idea of intravitreal injections, is that right now at end-stage disease, the only place the cone photoreceptors are left are in the very central part of your retina. This is known as the fovea. Right now you are using your fovea unless you have some type of retinal dystrophy. This is the highest concentration of cones, and this is what is left. So when you use the vector that we're using for intravitreal, that's exactly where it goes. And they have great transduction of those remaining cone cells. So this is basically what we are working on right now with these two programs in the gene therapy space. We are actively working on the CRISPR programs as well. This is just a picture of where we are. Unfortunately, I have the burden of coming to Paris about 8 times a year from Philadelphia. It's a tough job, but somebody's got to do it. And then, of course, we have our home office for myself in Philadelphia. So happy to answer any questions. Just catch me at the end and be happy to chat further. Thank you so much for your time.